thank you very much for the opportunity here, and Nina and Robert, to offer me to talk a little bit about what is going on in our institution as well as in U.S. In general scientific community. So I'm keenly aware of the 35% of attention retention span here. So in case you are going to fade out or pass out for the rest of my talk after 10 minutes, here's the bottom line. So genome editing tool these days you hear from literature as well as a newspaper are very, very powerful. You can deliver non-DNA. You can deliver protein-based, RNA-based, and I see my colleague in this institution is doing great on this aspect. And I will show a few current discussion topic on the global regulatory forecast. So in the end of the talk, about 10 minutes-ish, you wake up and you will see what I have to offer about this new regulation atmosphere, climate, in U.S. at least. So, okay. Let's start with the basics of the genetic diversity. To us, agriculture, that is extremely important that we keep genetic diversity. If anybody is saying that current large-scale agriculture or genetic transformation is to reduce the diversity, I think this is what we try to avoid. And then genetic engineering, and by all means that we try to keep and then even develop a further diversity for the crop improvement, because we know keenly is that for the future development, we have to use diversity as a source for the improvement. And we know we have been taking advantage of a natural variation, in this case, point mutation or deletion, which occur on a daily basis. And that's our fundamentals of crop breeding. And we go out and then look for the mutation, and which are caused by natural influence like sunlight, UV rays, and radiations, and cause those genetic variation. We take advantage of that, and we select them, and then that's our crop breeding. Those type of genetic breeding, natural breeding, in the late several centuries become not enough because our population increase exceeded of the food we can supply. Therefore, the new modern mutation technology has been adopted for several decades now, as the FOA data release, with about over 3,000 of species, all the plant material on the marketplace is coming out. It came out from those type of mutation breeding. As you can see, this is Institution of Radiation Breeding in Japan, and what they do currently, what they are doing is that one nuclear reactor in the center, and then different rice variety is planted systematically alongside of a radiation of the reactor, and then they were screening for mutations at a different distance locations. So this is how Japanese breeding rice these days, and they are not accepting transgenic, but they are doing massive mutations there to come up with new varieties. And those DNA changes and can range from a few as a 4 base pair to 8 kilobase deletion. You can also find inversions through this type of mutation breeding, insertions and frame shift, you name it. The key is how do we screen all these material and find the one which were appropriate for our agricultural practice. So this is what happened on a daily basis. We pick up those mutations occur naturally. Those random DNA breaks caused by chemical and radiations, every organism has ability to repair them. We, on a daily basis, our cell receive those mutation inference, but we able to repair and restore the gene replication. However, though, and in certain time and during certain period of time, our body is no longer to repair everything accurately, especially when we are getting old. That's why some disease occur, and the aging disease related to aging disease, or cancer is one of them. So it's because of our cell repairing system have faulty or get less efficient in repair. Therefore, sometimes instead of restoration, you have disruption. And for the current genetic transformation or current genome editing process, we are actually trying to characterize the disruption part of the gene repair in the cell and to take advantage of that for plant breeding. So natural homologous, all non-homologous end joint repair system are not very high, and it's quite, when you look at them, those homologous recombination occur is rare in the eukaryotic organism. And however, the recent technology development using this, take advantage of this double-strand break and use nuclears or engineer the nuclears can raise very high the rate, and in here indicating it's about almost 1,000-fold increase at the natural repair system. This is where we are using currently for the last few years. Okay. During the winter of 2012, a group of scientists at Iowa State University decided to get together and apply for a big grant from our university president's multidisciplinary research consortium investigation. So basically, in Iowa State, we recognize we have a very strong cohort of the plant scientists who at the time was very much focused on talents. Talents, it's a previous nucleus which identify from Sensomonas bacteria, because we have a very good plant pathologist who is studying actually the fundamentals of talents. And so we patented, we actually have a very strong patent position at that time. And for the talent technology, we decided that it's time for us to unite behind the technology and develop a further platform of research. So we formed a group called ICU Crop Bioengineering Consortium, and I served as a co-PI for that. And then we got over $1 million US from our president, and then give us, it's a 3-year grant, and allow us to start to network as well as developing technology, further develop technology and collaboration around the world to forging into this technology. Okay, our mission statement is to deploy innovative, transformative genome engineering technology that identify, validate, and rapidly but precisely integrate strategically important trait and underlying gene into key crop plant. And that time, it was the winter of 2012, and you don't hear CRISPR that time yet. We have not heard CRISPR. We only know talent is the best game in town, and we got the best technology in almost entire university, entire country. So therefore, we bank our money completely on talent technology at that time. However, in animal research, we start to realize that CRISPR is getting into more and more noise that time. However, 3 years later, and actually just last month, we offered a work called CRISPR workshop. Our university is completely switched from talents to CRISPR. Even talents is our intellectual property, and we changed everything, the full stream into CRISPR, because CRISPR is so easy and so highly efficiently we can incorporate for our crop of interest. So last month we have a very successful hands-on workshop, and then there are people from globally from many countries and attend this hands-on workshop, and we teach the student attendee how to identify the gene and then make the construct to perform a mutagenesis, and how do we do the maize and soybean transformation, and how do we do the genome editing analysis. So if you are interested in any of those, feel free to contact me. I can hook you up with the appropriate person, because our mission is to disseminate technology and help as many people in academic research as possible. So start a little bit of a case study of the talents. Dr. Bing Yang is one of the patent owner of this talents technology. It was his PhD study, and he was one of the first people who identified the talent come from of Sensomonas and isolated and identified the site-specific target in the plant. So in 2012, he published a biotechnology, Nature Biotechnology paper, and demonstrate first time the talents can be used to edit a rice gene and to convert the rice gene to become resistant to the disease. So what he has done is that one of the rice gene, OS sweet 14, apparently is a susceptible gene which was turned on by the bacteria. So bacteria is very clever. It hijacks plant system. So when the bacteria gets into plant, it spit this enzyme into plant and then make a plant switch on certain gene, and the subsequent of that gene switch on and cause plant to be susceptible to disease. So once you understand the mechanism, he decided to engineer the promoter of the plant sweet 14. And after the genome editing, the bacterial Sensomonas can no longer recognize the promoter. The promoter had changed. The bacteria gets into that and say, well, I don't find my target anymore. So the plant, consequently, the plant becomes resistant. So for many of the disease project, I think we have a very good future as a plant scientist, plant pathologist, as long as we understand the mechanism. Because we all know when we go to the field, we can identify certain resistant plant. And then if we understand what makes those plants resistant, we can really switch on the gene or shut down certain gene, and to use genome editing technology to make a plant become resistant. So this is a very good example. So genome editing for disease management or control in the future is very, very bright to me. So after this talent demonstrated very well in rice, we decided to switch on corn. And living in Iowa, everybody knows that corn is a king there, so you have to work on the corn. Rice is too unimportant for people in the Midwest. So we picked up a corn gene, which also tried to demonstrate what's the power of genome editing, and we choose the gene called GLASI2. It's a very classic gene that if you mutate that gene, and the leaf surface will become very different, because that particular gene involved in the deposition of the surface wax layer of a juvenile plant. So if the gene is mutated and the juvenile plantlet have very different surface reaction towards water. So again, we find a target region of the one of exon, and then we use talents to target and then make the mutagenesis. So this is the phenotype. After you mutate the gene, the plant leaf no longer repel the water. And because the wax surface layer disappeared, so the water just stick on that, right? So here is a wild type, and here is a mutant. And of course, those type of phenotype is not agronomic trait improvement, rather it's a reverse, because these particular phenotype actually is bad for plant, and you don't have a wax layer that allow you to repair the disease and all these the bacteria. So this is a really bad engineering, but we just try to make a case. We try to say, oh, well, we pick a gene, we engineer it and mutate, and then ta-da, very easily you just have that happen. Just shows how easily you can destroy a gene, or in this case, show the phenotype. So that was early on project. And then we switch immediately into CRISPR. As the minute we realize the CRISPR is much, much advantageous, and we start to jump into it. And then in a few months' time, you probably will see one publication from us that we worked together to establish a genome editing system in Kong. So this particular project aiming at establish a public platform for the community. And Kong CRISPR system has published probably a few of them, but both of those publications, their frequency is not very high. So in our case, from the design of and the construction of the CRISPR system, Cas9 and gRNA, and to the minute that you get a seed which have this edit plant is about 7 months, which is pretty fast for academic turnaround time. And then we have a software posted on our website allow you to identify the preferred target site. So all these things will have a reagent provided for you. You can either work with us and have us construct everything for you, or you can use our advice and then make construction for yourself. So far, we only have a limited plant species. In the future, we want to load up with all kinds of plant genome information, because you need genome information to design your CRISPR system, right? So the advantage of our platform is really high frequency. Here, we validated, in the publication, we showed a 4-gene example, in this case, the frequency. So the 4 gene is actually the 2 very similar gene in Kong, which is responsible for a pollen formation developmental process. You have called ZM-AG18, A and B. So this is a pair of a gene there. We decide to knock them out at the same time. We do it individually, and then we also do it in combination. So here that we identify the 23 transgenic callus culture, and among them, we have 17 of them has edits we want. So the frequency here is about 74%. So this is much higher than any of the published data so far. And then we go on and continue regenerate callus, become plantlet, and then most of our added callus line in the end of the day are positive in the plantlet as well. So this process also was proved or validated by 30 more constructs which come from many different universities, use our service system and generate that. And so it's a very robust. The technology, the design of reagent and so far. So basically, in Kong, in the case of a crop that you can do progeny, you can do transmission genetics, that you can receive those edits very quickly. So for example, here is a wild type, and you introduce your CRISPR-Cas9 reagent as a transformation process. So you get a transgenic plant, which carries your reagent, but you also have the edits caused by the CRISPR, right? So you will cross this particular transgenic line with wild type. And after this cross, in your next generation, you have a 4 different progeny population. You have a 1 carried your transgenic and the edits as a parent, and then and a mother parent, and then you have the population which carries only your transgene, And then you also carries the wild type, there is nothing there, and then you have a most important group of the population that is carries no transgene but edits. These occur in one generation. This is T0, we all know that. And then you cross it with the wild type, and then in one of those progeny, you will find whatever you want. So this is absolutely free of transgene, but has your edits there, which is fantastic and very fast that you can achieve what you want in one generation. So, and then I switch gear to the protein delivery, and then why do we care about protein delivery? Why we want to have a DNA-free delivery technology? So basically, you don't need to generate a expression line, and if you have a protein, you can immediately bulk up and then shoot them, and then no need to segregate progeny because you don't need to worry about transgene integration. And transient activity could reduce toxicity or off-target mutations, and Cas9 indeed, if you linger around in the genome for too long, sometimes they generate undesired off-target mutations in the genome. And of course, save time analysis possibly. So basically, this is a traditional approach. When the traditional approach is a transgenic genetic transformation, you have a wild type of clone, and you deliver your enzyme or DNA through enzymes through a DNA format, either agrobacterial-mediated transformation or particle bombardment. So here, then you will have this transgenic line carries your reagent as well as edit plant. And then the next generation, just like I mentioned earlier, you will have a segregation, and you will have this undesired and then population, but you can figure out and then find the edited plant. This is our traditional way of doing for the genome editing. However, if we choose to use a protein delivery in this case, what our approach was, we put into the protein into the nanoparticle, which is porous in nature, and then we bombard them into plant, and during the transient expression, they will create edits in the plant genome, our desired edits, and then these are immediately we can acquire a non-transgene bearing plant which has edit, in this case, a mutagenized plant. So this is our desire, which we started this approach actually as early as 2007. That time, we already had interest to carry out this work because we had a collaboration with a chemist on campus who is expert in the material synthesis. So he synthesized a mesoporous nanoparticle, which is highly regular particle and very stable, thermal or chemically in this case, and they have a very large pore, tunable pore size, allow you to put a large of a payload there. And then also very important, you can surface conjugate, surface decorate, functionalize material, allow you for different application. So it was this type of a collaboration that generate our interest of using this material for agricultural application. So here again, that these particular molecule can be filled with chemicals or molecule with simple diffusion, and then they can be covalently linked on the surface and can be used for release under a different condition once the surface can be open release. The reason when we start this approach is because at the beginning we realized the silica porous nanoparticle is impossible to be introduced into plant cell. And we don't want to work with protoplast because we know for protoplast, corn and soybean transformation will never get anywhere because these cannot be transformed and regenerate through protoplast. So we know we have to prepare these molecules into plant using BioListic that time. And then silicon nanoparticle was too light for us to achieve that. So to circumvent that, and my chemist partner decide to coat the surface of the nanoparticle, in this case silica, with a small gold to add weight to that. And 2007, we co-published this paper, and it was, titled here called Plants for Change, because for those of you working in the nano material area, you hardly hear anything from plant side because everything is a cancer cell and human cell, and it's much easier to work on those material. And so that's a seminal work for the plant side of utilized nanoparticle. And these are the commentary paper just shows potential application for nanoparticle in the future to introduce non-DNA based molecule, or to introduce multiple molecules at the same time. So right after that, we received the interest from industry, and they want to work with us for the delivery protein. It was very obvious that time that we want to introduce non-DNA molecule into plant in order to achieve the type of immunogenesis, and that was 8 years ago from to date. And so immediately we were just saying, okay, can we encapsulate protein into pores? And of course, a nanoparticle at the time was not big enough for us to encapsulate large protein, but later on it was modified, and so we can actually encapsulate GFP in this case. And note, we have developed a protocol to gold plate the MSN with gold. Gold plate 3 times, and not only increase the weight, but also increase the weight of the surface character, allow us to introduce a protein a little bit easier, which is very good. And here you visualize a GFP delivered onion cell, and later on we co-deliver a protein with DNA, and we can characterize expression of the DNA, and here is the alpha P in red, and co-deliver our GFP as a protein and same cell. So this was just shows that completely possible for us if we want to make this molecule applicable for the plant delivery. So of course, the ultimate goal for this pioneer collaboration was not to just show how pretty the GFP alpha P is, but rather to demonstrate actually you can use this to deliver enzyme or recombinase in these early days to see whether that can be functional even during the introduction to the plant process. So here that what we have been using is a material which is a Cray protein, which purchased from a commercial source. And then the LOX cone, which is generated by a Pioneer group. So that piece of a LOX cone, the transgenic cone, we use as a model, as a case study. These particular LOX cone carry a 1 copy transgene. That transgene carries a promoter, drives a EPSP glyphosate selection marker, and another promoter drives a SINE YFP CFP gene. And then you have a DS red without any promoter. So the idea, and then in between these 2 fragments, there's a LOX P site incorporated here. So the idea is that if we introduce a Cray gene, Cray protein into the plant, if the recombination does work, these fragments will be deleted. And the promoter here will be driving the alpha P, DS red, and to make the cell turn from blue to red. So that just, the Cre-Lox is at works, and then you will see the consequence of the recombination. So you basically will see from a blue cone to the chimeric red and blue cone. So, and indeed it's a case. So at early days of transient, you can see the blue channel, those cone embryo was completely blue, and then after 2 days you will see the specks. Actually, not after 2 days, after really very short period of time, 5 hours within a day time, you will see red specks indicating excision of recombination already is taking place. So then afterwards, you can follow the red appearance of the callus culture, just shows the proliferation of the transgenic and recombined event. And this particular cone callus shows half and half. You have a recombined sector and out of this blue original parent callus culture. So these frequency actually pretty high, like 20% of that. And these are the shows also the product of transgenic and recombination event. So when you isolate all these red stable plant and then you perform the DNA analysis, you will see the excision, precisely the excised and recombined site, very, very precise from all the event you analyze. So that was a very good example showing, well, you can do it. You can introduce functionally important enzyme into plant and then make the work, and the functionality retains. And it's very nice to demonstrate that. And then, as I said, that the frequency is about 20% of the recombination, which is very, very comparable for all these published data using DNA as delivery. Here is a protein delivery, and then when you deliver as DNA, you will reach a similar frequency. So protein delivery is not to any disadvantage in this case. So these are all good. Signs are there, and what happened to regulation? So when we started our consortium, the first thing we would just say, oh, wow, these genome editing tools are so cool. How did regulatory agency will react to that? So Dr. Yang mentioned that I have these talent strain which are completely free of transgene, right? So he had 7 talent-generated disease-resistant rice line. And first he did 7, and everything shows there's no more transgene there. Great. And we will say, okay, let's have a genome sequencing again. So he sent the 7 line to get total genome sequencing again. The purpose is to see, do I have any transgene left in these lines? And come back and says 5 of them is completely transgene free, whatsoever. Everything is the natural rice gene. But there are 2 lines, even 7 was negative and show some residual transgene there. Then we sent out an inquiry letter to the US APHIS, which is a regulatory agency for all these transgenic research. Because we don't know if we want to apply a permit or not. There's no guidance from anywhere tell us what to do. So we write an inquiry letter of February 17th. We wish to receive it early enough so we can plant in 2014 summer. Well, we didn't receive until a year later. When we keep calling them, we say, well, and 2014 we were nervous because we want to plant them in 2014 summer field. And we didn't hear from them, and then we call them and say, well, what's going on? They said, well, we don't know. They have no idea what to do. So a year later, we received a letter from them, and one of the sentences says, based on the information cited in your letter, APHIS has determined that the 5 rice line were developed use plant pests and genetic material from plant pests. However, the final rice plant do not contain any insert genetic material, and APHIS has no reason to believe that the plants of this rice line are plant pests. Therefore, consistent with the previous responses to similar letter of inquiry, APHIS does not consider the 5 rice line as described in your February 17, 2014 letter to be regulated under 7 CFR Part, and APHIS has no reason to believe that transgenic genetic engineering of your GE rice would increase weediness of rice. What that mean? That means we don't care. You can do whatever you want. So basically, in this case, it's a very interesting case that we all learn from that. They did consider the entire process. I will talk about this process versus product in this entire thing, and which is very confusing, and which is something that we have to face all the time. Okay, this year, last month in April, we read from a Science that next generation GMO food is here. That's technically not GMO, which describe a DuPont Pioneer high-quality waxy corn. Is basically it's mutagenesis of a waxy without any transgene using the CRISPR technology, and then increase the digestibility of the waxy, which is one of the very much wanted industry trait in U.S. And then you all hear this gene-edited CRISPR mushroom, right? So these are from my friend Yinong Yang from Penn State, and we all start our life, our research for fun. We just say, oh yeah, there's a gene we can knock out, and we knock out. Actually works, and the mushroom no longer brown very easily, and that's a fantastic product, right? So UPenn brown resistant white button mushroom. Then we hear all this nice, it's a good news for biotechnology. Oh, it's a reason for public concern, because look at all these headlines. Dodges, bypass, escapes, they're all have a negative, condescending feelings about, oh wow, they are as bad as GMO, just regulatory is no longer have anything can control them anymore, right? So this when for those people who does not like GMO, this technology does not make them feel any better. And we thought, oh wow, that new generation of a genome editing will help us to move forward. Are we able to? Okay. So genome editing methodology catalyze a double strand to a specific location of a genome. This is what we know. And then we know there's a bunch of technology available. And I started this genome editing as early as 2003, because all our molecular biologists, most of senior molecular biologists, we know our ultimate genetic transformation, we want to be precise. We want to go to the place that we want to go. We don't want to have a random insertion and everything. So we start the zinc finger nucleus. We've gone through all different type of things, and then now CRISPR become completely possible and so easy, okay? However, how much regulation will severely impact the future success of R&D of this technology, which we call GENE. It's genome editing with engineered nucleases. There is a conundrum for this particular technology. So for those of you, English is not, for those of me, people like me, English is not the native language. What is a conundrum? Conundrum is a confusing and difficult problem or question. So which means if you ask your regulatory agency, everybody was like, huh? What do we do here? It's not as easy as we scientists think about it. We think it's so straightforward. I demonstrate it's so straightforward. There's nothing wrong with the technology. But for them, there's technical issues. What differentiates a gene from the other technology developing plants with unique trait and phenotype? Oh, the process versus product conundrum, and is a concern of the genome editing of the derived phenotype. So, case study. We know there is those low-phytate cone out there. Phytate is a very important. This low-phytate trait for cone is very important for the livestock industry because when you see smell all the bad manure and so on, it's because they ingested, they cannot digest certain things and cannot absorb. So low-phytate cone is basically try to reduce the pollution and then enhance more absorption of a nutrient, right? So this product has been very much demanded by industry, and you have multiple ways of doing it to reach the low-phytate cone trait. So you can use the reagent of a different way. You can have traditional mutagenic method or recombinant DNA technology, which is a transgenic, and then now you can use genome editing to reach that. Okay, what have we have? So for traditional, of course, you don't need anything, just go screen, right? Go to the field, screen. And then you have mutagenized, you can use this chemical to do that, or you can overexpress a gene of interest, and then these actually be accomplished by using zinc finger nuclears already exist. So the result of this treatment is those low-phytate maize with phytic acid pathway via phytic acid pathway. So it's all possible with different ways. And then result of a regulation. For traditional, you don't have regulation. Mutagenous, you don't have regulation. Transgenic, of course you have regulation. What about genome editing? Maybe. We don't know. So when you look at them, the more familiar you have is less specific in this case, right? This technology, the more specific and less familiar with people, and people worry most. The more the science improvement, the less people have confidence in you. What's wrong with our society, right? You know, we need to teach from a grade school on to have people have a confidence in science. It's opposite, seem to be. And then you have this process versus product conundrum, okay? Canada is the only country which don't care about whatever you do to reach the goal as long as your product doesn't have a problem, has a trait that you intended, doesn't have a side effect, they will approve it. Many countries, Japan, EU, to name a few, are the process-based regulation where they care about whatever you do. They care about, okay, does that include technology? As long as you include technology, they want to regulate you. And US has a split personality. So, and sometimes it regulates, in our case, for example, it's obviously and regulates both product and process. They worry about, do you use agrobacterial transformation? Yes, we need to regulate you if you use agrobacterial. If you bombard, no, it's okay. Seriously. So that's why bombardment is still becoming really important technology, because you tell them that it's biolistic media transformation, they don't regulate you. Okay? So technique here is genome editing, cisgenic, transgenic, mutagenesis, traditional. You use a reagent of a CRISPR, Cas9, or TALEN, etc. And then you reach those goals, which is genome editing, right? Then in the end, it's a unique phenotype you want. So these are the process, and then these are the product here. So these, exactly what I'm saying, that if, as long as EU and Japan continue to regulate process, genome editing will not receive less regulation than transgenic. So here, regulatory significance for genome editing. If you use direct introduction, which means not DNA, but protein or RNA, and you reach those type of the product, which is indel and deletion or familiar insert, you are likely to receive no regulation. In our case, we had that. Okay? And if you introduce novel inserts or transgene inserts, of course, they are going to regulate you. If you use DNA for introduction of your transgene for CRISPR reagent, but you will do the progeny segregation, and you will receive different treatment in US and in EU in this case. EU will still be worried about it, even your next generation is free of transgene. Okay? If you have a transgene in the progeny, you will be regulated all over again. So, what is the global climate for the future? It will be partly or mostly cloudy, and basically means we have no idea. Okay, DNA insertion with NS is new segregant selection. Just I mentioned that you introduce reagent, but you segregate out next generation. And in US, it's okay, good. We already have many cases that can be done. Canada, of course. Argentina, yes. Australia, just most recently. Join the rank of the sand people, this to me, and Sweden. And then you have a DNA insertion with any stage. New Zealand, your friend here. And then Germany, most advanced in technology, but Germany had a catch. Say, well, if you directly deliver protein for that, that's fine with us. So this is something which is you take into consideration. It's not straightforward. If you will my transgene genome edit crop to be subject to regulation, maybe. Unless there is a clear regulatory precedent, you really can't know, and even then you cannot know for certain. So this is what we are facing here. And what that means to us as a scientist. It's very important for us. We keep doing the research. We keep experimenting. If we don't do that, we never will have answers. So we need to continue optimization and then validation of the specificity, which is very important because right now, sometimes you use CRISPR to target your transgene, fine. It works very well. And it goes to 10 different off-site as well. And that creates a lot of concern from people. And you need to improve your bioinformatic production. You need to have a more of genome sequencing information. And then you need to address what's this arrangement and multiplexing all these things. And also, you need to be very worried about research on the gene drive. That people know gene drive. Gene drive is one of the things which right now are very much talked as a climate of Zika virus, right? So you have those transgenic mosquito released to the environment with the intention to wipe out all the Zika carrying mosquito. It's a good thing for us. Is that really? We don't know. So with a gene drive inheritance, the American National Science Academy caution really severe cautions small field test not to just release out as it is. And it can have a really big consequence for that. Because for those of you who may not familiar with gene drive, which is basically if you have a mosquito carries CRISPR reagent, which will target the Zika virus carrying all the reproductive part of virus, the mosquito, when the CRISPR mosquito mate with natural Zika carrying mosquito, next generation, it will most likely be modified to carry more CRISPR gene, and then they will go on and then target more mosquito. We make the population non-reproductive. You wipe out all mosquitoes. That's okay with me, but you know that. So here, the potential application with a gene drive is quite appealing. Here you can have a public health advantage, which I already alluded, and a lot of those human disease come from a mosquito. And then you have also ecosystem conservation, and you can eliminate some of the organism which has invasive nature of that. Then you have agriculture application, and you have, of course, basic research application in this case. But to carry out this field release or outdoor or whatever, it has to be really very carefully planned and evaluated before we carry out, to my opinion so far. To close my talk here is that, okay, our research has a lot of a risk, looks like. Even we do all our best to achieve the as little of risk impact into society, but we still will be facing every day, and we've considered every potential risk except the risk of avoiding all the risk. So thank you very much for your attention here.